Okay, well, I'm afraid I just had a disaster with my computer. My computer crashed. I was two-thirds of the way through the class and that I was recording, and my computer crashed. So I'm unsure what to do. I'll see if I can find what I had been recording and post that. But I'm just going to repeat where I was when the crash happened. I'll repeat this section and try to get as far as I can. If need be, I will re-record the first part of this lecture and put it up tomorrow. It's Tuesday. I apologize. I do not know why on earth it did that, but it did. So let me just tell you first of all. Let me see if I can get my what I was busy talking about was the infection of bacteria by their own viruses, and the first that we have a look at are the T-even bacteriophages. These very complex viruses, and these are very common in bacteria. They're a common way of controlling bacterial populations, and they can in fact be used therapeutically as well. They has been done in several instances. So I will emphasize that these all stages, which we see in all viruses, here we're referring specifically to one group of viruses infecting bacteria, but we see the same stages of infection: attachment, penetration, biosynthesis, maturation, assembly of virus, and release of new viral particles. We'll see that in all viral infections, no matter whether we're talking about bacteria with their phages or us with our own viruses. So I want to describe, first of all, that in bacteriophages, there are different ways for the phages to relate to the cell. And that is something we see in other viral infections as well. In this case, for example, this phage, T4, the T-even phage, causes what is called a lytic cycle. So the phage enters the host cell, injects its genome, which is DNA. That DNA immediately cuts up the host genome, keeping only those parts which are useful to the virus for its own replication. It doesn't destroy the entire cellular machinery because it needs to commandeer the functioning. It needs to commandeer especially replication to replicate its own genome, and then the translation machinery so it can produce its own, the viral proteins. So that's exactly what it does. It effectively turns the cell into a viral factory. The viral proteins are produced. Once they're produced, they actually assemble themselves into the new viral particles. Once they're assembled, they package the genome. They package the genome which has been replicated by the host machinery. And so they form this maturation phase, they form mature viral particles. Once that occurs, the cell is induced to lyse, usually with a set of enzymes like lysozyme, which burst the cell and release the viral particles to go on then to infect other cells. And I'll just mention, in case I never get the split back again, I'll just mention that viruses don't always establish a successful infection in bacteria. It can happen that the bacteria are able to resist a viral infection. And they usually do so by chopping up the genome with their own restriction enzymes, which recognize the viral DNA is present and cut it into pieces. But really importantly, bacteria can keep small pieces of viral DNA as a record. as a memory of a previous infection that has been successfully overcome, so that if ever they see that DNA again, they are able to very rapidly destroy it. And this is important because this is the basis of that of a system of genetic engineering, which is dependent on this called CRISPR-Cas9, which we may discuss in a little bit of detail later. Okay, so this series of events here leads directly to the lysis of the bacterial cell. Now, that is not necessarily for all viruses the way in which they proceed. There are many other viruses in which the virus actually adopts a different strategy, and for a time at least, integrates its genome with the host genome. And this is referred to as a lysogenic cycle. One of the most famous of these is this other bacteriophage, Bacteriophage Lambda, which undergoes, it looks very similar to T4, also a DNA virus. But when it injects its DNA into the host cell, it doesn't immediately destroy the host genome. Instead, it integrates its own DNA into the host DNA. There it is there, and that we refer to as a prophage. Significance of the prophage is that whenever this host cell replicates, whenever it divides, it also replicates the prophage. So you can end up with many, many, many billions of copies of the prophage present in a population of bacterial cells before the virus actually replicates itself at all. This can go on for some time. But in response to particular cues, which vary, it may be that the cells become stressed due to lack of nutrient, or that they suddenly get a new burst of nutrient or something. But some environmental cue cues the virus to actually leave the host genome, and it forms its own, it's like a small chromosome. It looks almost like a plasmid. So it exits the host genome, forms, and now enters into the typical lytic phase. Once this viral genome has left the prophage stage, it commandeers all of the cellular machinery. And just as before, just as we saw in the lytic phase, it forms new viral particles and causes the lysis of the cells, and then carries on with the infection phase. So it's just that there is a period here of latency, which can be very long. And in fact, we frequently, if we look at bacterial genomes, very often find, located in their genome, we find prophages which are sitting there completely silent and would only come out in response to very specific environmental cues. This is important not only for bacteria, but it also points to the fact that in many human viruses, especially the herpes viruses, a similar kind of strategy is employed, although of course our genome is completely different. But they are also able to enter a long latent stage and only emerge in response to very specific cues, physiological and environmental cues. So that is those are the bacterial viruses. Now, what about the animal viruses? How do they differ? Well, in actual fact, we can use the bacteriophages almost as being analogies for animal viruses. First of all, animal viruses, in order to establish an infection, they need to attach, and they need to attach to the correct cell. They need to attach to the cell membrane, and they will only attach to cell membranes which are correct. And the appropriate cell membrane is identified by the exterior proteins of the virus. If it's an enveloped virus, by viral proteins embedded in that envelope. If it's an unenveloped virus, by proteins in the capsid. And those are, certain of those proteins are recognition proteins that are able to bind to specific proteins in host cell membrane. If they find the correct one and they are able to attach correctly to those proteins and to the cell membrane, then they gain entrance. They gain entrance to the cytoplasm of the cell in one way or another. There are two methods for them to gain entrance to an animal cell. The first is to be taken in an endosome, as if they were being phagocytosed by the cell. The second is to merge an envelope with, or capsid, with the cell membrane and open up the cell membrane to enter the cytoplasm. So the one is by endocytosis, the other is fusion. Once inside, remember that these viruses have a capsid, which is entering the cytoplasm. The capsid has to be removed in one way or another, and that's called uncoating. Sometimes done by viral enzymes, which it has with it inside the capsid, or else by host enzymes. The proteins, the viral proteins are able to induce a reaction from the host enzymes that will digest them and liberate the genome. A lot now depends on what that genome is. If it's DNA, it's usually integrated with the host DNA. If it is RNA, there are various strategies that we will talk about. But ultimately, the viral genes are utilized by the host cell to produce viral proteins, and also the viral genome needs to be replicated in one way or another. Again, just as in bacteriophages, once all the viral proteins have been correctly made, they will assemble and they will mature by packaging the genome. And once that is complete, we then move to the stage, very important stage, because the viruses need to get out. In many of the animal viruses, release is by budding. Some viruses do cause rupture of cells, but by far the majority in animal viruses, the virus actually buds out. The advantage to the virus is that the cell, the host cell, remains viable for longer. In addition, we could think that it may be advantageous that it doesn't excite as much immune response as you would have when you've got bursting cells or dying cells, which are releasing substances that excite the immune system. So here are the two methods. The first is endocytic route. That means it is taken, the virus is taken in by endocytosis. This, by the way, is something like an adenovirus. It's unenveloped, but enveloped viruses can also be taken in by the same method. And indeed, our coronavirus, an enveloped RNA virus, is taken in by this method, by endocytosis. What happens is that the viral proteins bind to specific signal proteins on the outside, and their binding induces the host cell to form an endocytic vesicle. Here it is here. And that the virus now needs some strategy to escape this endocytic vesicle. Very often the endocytic vesicle the membrane simply breaks down, but we'll see, I'll show you for coronavirus that there are other strategies that can be used. Ultimately, the capsid is released, digested, and the genome is released. This is a fusion route. This is quite common with enveloped viruses because this outer membrane, the envelope, is quite similar to the cell membrane. Once the viral proteins bind to the correct signal proteins on the host cell membrane, the two membranes unite and open to kind of spit the capsid of the virus into the cytoplasm there. Here, these are electron micrographs of this happening. Here's an endocytic vesicle forming to actually take this viral particle in. There it is here. And here is, this, by the way, there's, this is the envelope around here of this particular virus. There's the capsid there. This one here, here's the envelope here, and here the capsid on the inside. And here, this membrane here is uniting there. And this will open up to spit the capsid inside the cytoplasm. So it's, at this point, is actually a good point to mention that similar processes can lead to the release of virus. So, for example, very frequently, that envelope that we see around enveloped viruses is acquired by budding of host membrane. Now, I'm being very careful here not to say cell membrane, because in fact, the viruses may bud out and acquire their envelope from any of the host membranes. It depends on the virus, it depends on the viral life cycle. Some viruses acquire their membrane from the nuclear membrane, others from endoplasmic reticulum, others from Golgi apparatus, some from endoplasmic reticulum and Golgi apparatus, which is the case for coronavirus. But here you can see how, in any case, here a capsid is inducing the budding out of the virus with its envelope around it. Very often, usually, in fact, the virus, before this budding out takes place, will induce the cell to plant viral proteins inside the membrane, so that when the virus acquires its envelope, the envelope has the requisite viral proteins embedded in it. But remember, this can happen at any stage, depends on the virus, on their life cycle. It can happen budding out of the nucleus, endoplasmic reticulum, from the Golgi, or from the cell membrane. HIV, for example, acquires its envelope from the cell membrane. Coronavirus from endoplasmic reticulum and the Golgi. Okay, so I'm not going to, at this stage, talk that much about viral disease, but I do want to mention these special circumstances of viral infection, which can result in cancerous outcomes. And we know that several different types of cancer are caused by viruses. I have previously mentioned things like the papillomaviruses. The papillomaviruses can cause cancers of the genitals, for example. They can cause cancers of the cervix, can also cause cancer of the penis, along with the fact that they cause an immediate, acute, more acute disease, which is warts, genital warts. So these cancers resulting from viral infection may develop long after a viral infection is finished. The cancers themselves are not contagious. They have long since passed the stage where the virus is actually being released as viral particles. And instead, the virus has induced changes in the behavior of the cell. It's easier to understand it like that. We know of several of these sarcomas, diseases of connective tissue, adenocarcinomas, they're cancers of glandular epithelial tissue, and several others that result from. We refer to this process as transformation. And this is transformation of normal cells to form tumor cells. It's a different use of the word transformation. You'll remember we talked about bacterial transformation was inserting new DNA. This is actually changing the nature of the genome of the host. And the commonest mechanism for this to happen is by viruses acting on genes in the host genome, which are referred to as oncogenes. The oncogenes are those genes which have the potential to produce a cancerous or tumorous situation if their function is altered. I'll give you an example. Cell division is very, very carefully regulated by numerous mechanisms. Cells only divide at certain very specific times. And there are many mechanisms for the cell to regulate its cell division. One of them is, for example, that the cell responds to growth regulators. So substances which are in circulation, there are certain factors which will cause cells, when they see them, to begin the process of cell division. Now, to see them, for example, cells have receptors in the cell membrane which bind those growth regulators. When the growth regulator is present, binds to cells' proteins in the cell membrane, and that sends a signal to the cell. It says divide. So here's the problem. Here's one of the ways in which viruses can affect this. In a system like that, a virus inserting nearby or into that regulatory gene inside the cell can turn that gene on permanently. So the gene, this regulatory gene, which normally only responds to the presence of the growth factor, now starts to produce its product the whole time. That is a typical situation where a virus has inserted near or into a regulatory gene and switches it on permanently. That cell now loses all its control, all its division control. It now begins to reproduce constantly. It reproduces over and over and over. And this is typical, for example, of things like the papillomaviruses. Now, in their acute phase, all that happens is the cells reproduce rapidly, and they produce what we recognize as a wart. But those warts are very soon recognized by our immune system and are destroyed. But certain strains of papillomavirus, inserting into the correct cells, for example, cervical cells of the cervix, can actually alter the function of oncogenic genes. And they can end up turning those cells into cancerous cells. And this may happen much, 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 much later, decades down the road, that this process actually commences. So they have the potential for making cells become malignant. And that is that the cells lose all growth control, but they also begin to lose their ability to associate closely with one another. They begin to acquire the ability to spread through the body and to begin to spread the cancer from a primary site to secondary sites where the cancer will continue to grow. So there are a number of these oncogenic viruses. And I'm sorry. These are a whole list for you of oncogenic viruses, which are DNA viruses. Adenoviruses, which often cause relatively mild acute disease, are associated with a number of carcinomas. The herpes viruses, there is one group, one strain in particular, which called the Epstein-Barr virus, causes a number of diseases in humans, relatively mild diseases, mononucleosis and those sort of things. But it produces a very distinctive cancer called Burkitt's lymphoma, particularly in people of African origin. The various of the members of the pox viruses can also be oncogenic. And then the whole human papillomavirus I have also already talked about. Now, this is another group, a very strange group of viruses called the hepadnaviruses. And they can cause hepatitis. It's a relatively mild hepatitis. In fact, it might even be asymptomatic in some people, but eventually it can cause liver cancer due to the oncogenic activity of the hepadnavirus. There are many others. There are hepatitis C, for example, can also be oncogenic and cause liver cancer. So that is the one long-term activity of viruses. The other long-term effects are due to the fact that viruses can often remain latent. This is particularly true of that group of viruses called the herpes viruses. And the herpes viruses, often they will cause an initial acute phase of illness, and then they'll go into a long, long period of dormancy when they actually remain silent until some environmental cue produces acute symptoms again. So the most important ones are things like the herpes viruses that cause mouth sores, cold sores, and also genital herpes. Both of these can become latent. Sometimes they go latent and they never reappear, and that is actually the most common situation. In other cases, people undergo a period of some years when they have recurrent outbreaks, and it then becomes silent. This disease here, shingles, is actually the result of infection by the chickenpox virus. And chickenpox virus causes usually a acute childhood illness, but then becomes latent for decades and can reappear as another disease called shingles. When we talk about viral disease, I'll mention it again. There's one disease, it is I would say it's relatively rare, but something very, very important to think about. And this is not a latent illness. This is a persistent viral infection. Now, some viruses persist for a very long time, actually in a low-grade infectious state. And this is a very important one. It's called subacute sclerosing panencephalitis. And it's a result of infection by the measles vaccine, by the measles virus. And the measles virus, in this case, actually carries on activity literally for decades in a very low grade. It's not latent. It is actually present. It can be found if it's looked for. And it then can cause this very, very, very dangerous brain infection much later on, decades after the childhood illness has occurred. This is one of the reasons why measles vaccination is so important. Even though some people regard measles as being a mild childhood illness, which it's not, it's extremely dangerous childhood illness. It can also produce this long-term chronic condition. There's a whole list for you. I'm not going to go through them all. You can have a look. Several of the things I've mentioned are here, but you'll see that this is latent, oncogenic, and persistent viral infections are important in human beings. Just to finish off, we talked about bacteria, we talked about animals. I'll just mention two smaller things. Plant viruses are incredibly important. There are many, many, many plant viruses. Plants are somewhat protected from viruses because they have a tough cell wall. But anything which breaches that cell wall can actually transport virus into the cells of the plant. And plant viruses are tremendously important for humans because they cause so many crop diseases. They're usually spread by insects. but they can also be spread by any wound. So pruning, this sort of thing, if gardeners are not careful, they can easily spread virus from one plant to another. There's another group of infectious elements called viroids. These are very strange. They're little pieces of naked RNA, only occur in plants. And they're just naked RNA. They have no capsid or anything. Again, they're usually transported by insects. Usually the infection is caused by a parallel infection with something like aphids or something like that. Just short, naked pieces. Viruses, all of these things are very peculiar. They will adopt any strategy, it seems like, in evolutionary terms to reproduce. Here's a viroid, just a little piece of, this is potato, spindle tuber viroid. There it is here. And the last thing I'm going to mention is another, you can't, I'd say it's an infectious element because it's just really a complex biochemical. And that these are the prions, proteinaceous infectious particles. These are misformed protein strands, misformed polypeptides, which once they get entrance to a cell or form inside a cell, induce all the other proteins of that kind to fold into this mishapen form, and they start interfering with the functions of the cell. And the best known are those, first of all, mad cow disease, which I'm sure you've all heard of, but in human beings is called Creutzfeldt-Jakob disease. It's also very important, can be very important, not only in cows, but in sheep as well. And we can acquire it from both of these. Unfortunately, the unfortunate thing about these prions, the protein-infectious particles, is that they're quite easy to acquire by eating them, acquiring them. They have been in transplants, and people have acquired them from transplants. It's very, very difficult to get rid of them by sterilization or even autoclaving. And so they can be transmitted by surgical instruments, for example. And they result in massive neurological damage. Now, this is not common. Don't panic. Think it's not. There have been outbreaks of Creutzfeldt-Jakob disease, mainly associated with people eating meat from cows that are infected. So with that, I'll finish. But I will go back and try and find the first half of this lecture. If I can't find it, I will have to re-record it and post it tomorrow. Okay, I apologize.